So, so we are almost there in the final session today, and the first day of this ATAL FDP faculty development program sponsored by AICTE, and on applications of CRISPR-Cas gene editing tool. So we had already 2 sessions prior to the lunch. One was delivered by Dr. Shovik Maiti from IGIB, and the second one was from Dr. P. Chandrasekhar from CCMB. So today it's all CSIR group, it seems. I just happened to notice it, right? Yeah, it just came out accidentally or incidentally. But yeah, incidentally. So welcome, Dr. Debobroto Biswas. So Dr. Debobroto Biswas is currently the principal scientist and the deputy head from the Council for Scientific Industrial Research, IICB, that is Indian Institute of Chemical Biology, which is in Kolkata. So CSIR, IICB, that is how it is known as. So Dr. Biswas is currently working on, as a principal scientist, as I've already mentioned. Before he joined IICB, he completed his PhD in experimental pathology from the University of Utah at Salt Lake, and from Salt Lake City, Utah as well, in US. So after completing his PhD, he joined the laboratory of Professor Robert G. Roeder at the Rockefeller University, New York. Then he moved to IICB Kolkata, and his primary focus is on the mechanistic understanding of transcriptional regulations within the mammalian cells and their implications in several human diseases, including cancer and type 2 diabetes. I think this session will definitely be a very, very interesting one to the audience. So work in his lab means the fundings that he has received so far is from a CSIR, from the DST, as well as from the Wellcome Trust DBT India Alliance Fellowship. And today, Dr. Biswas is going to talk on the CRISPR-Cas9, a tool for modulating gene expression for mitigating human health diseases. So welcome, Dr. Biswas, once again, and the stage is yours.
Okay, I hope I'm audible to everyone, right? Jayadi, can you please confirm?
Yes, yes, okay. Yes, absolutely, okay, very clear.
That's good. Okay, thank you, Jayadi. And well, I'm used to calling Jayadi and not Dr. Bandyopadhyay because that sounds more of professional and all these things. And let's not go into it. Okay.
Yes. And you can, and Debobroto, you can put it in the slideshow mode whenever you like.
Absolutely, yeah.
Yes.
Okay, there you go. Yeah. So let me start, and then thank you, Dr. Bandyopadhyay. And then let me start with my talk. And the title of that talk is CRISPR-Cas9 and its implication in understanding mechanism of gene expression and its implications in human health diseases. So what I understood from Dr. Bandyopadhyay is that the overall audience is from a diverse background, which is in a sense that it's good, but also at the same time it's a bit challenging in a sense that sometimes some of the jargons that we use can be a little bit of going off the top. And so if you feel anything, so please raise your hand, and I'll be happy to kind of address any question that you may have while I'm giving this presentation. Also, at the same time, this talk is right after having a fantastic lunch, whatever that we had, and then so keeping everyone awake is another primary challenge that I'm going to have, which I hope that I will be able to make it. Okay, now going into it. So since there are diverse audience are there in terms of and their interest in the CRISPR-Cas9, which is always good to see that the people from other area of expertise, they are also interested in this technology. So I would go starting from a very basic to the gene expression, and from there on the applications of the CRISPR, how we can use it for modulating the gene expression for addressing several diseases. So the first part will mainly be focusing on the gene expression and some of the mechanisms that we know. And then the next part would be mainly to use the CRISPR-Cas9 as a technology and how to address this, whatever the mechanism that we know in terms of mitigating the diseases. Okay. Now, this is a textbook, that images that you see with a human body structure, wherein the human body is composed of multiple organs, and all the organs have their own type of specific type of cells. Interestingly, all the organs also have, the cell that they have, every type of cell has, they have their own identity. So a given set of genes are being expressed in a given type of cells, in a given type of organs, and that's what makes it specific. And however, the underlying gene sequences are all same in all the organs that you would think about. So the question is that why a specific set of genes are expressed in a specific set of cells or an organs that ultimately define the organ, and then perform the overall day-to-day activities that we do. So now going into it, in the detailed mechanism of these expression of these gene expression in terms of addressing these in terms of gene expression, and if we see the every cell and then we cut it open, and this is once again the textbook knowledge that we see, and you can see the mitochondria, the Golgi, the nuclei, and all other different types of organelles are there within the cell. Okay, and one of those that we'll be focusing on today within this cell is the nucleus, because we are going to talk about the gene expression. Gene expression means genes which are encoded by the DNA, which is there in the nucleus. So I'll go from the human body organs from the organs to the cell, and within cells we are seeing all these different types of organelle. And the organelle that we are going to focus about is the nuclei or the nucleus. Okay. Now, if we cut open further the nucleus, then what do we see within this? There's a one dense, dense structure is there within the nucleus called the nucleolus, and within that, within the nucleoplasm, there are several chromosomes which actually harbor the genes that we are talking about, which would be expressed. Okay. Now, in human, we have 46 chromosomes. There's 22 somatic chromosomes, and then 2 sex chromosomes, X and Y, depending on what you are talking about. Now, within the chromosome, now you have the DNA. So what is the structure of the DNA that you see within the chromosome? So from a human body to the organs, to the organelle means we are talking about the nuclei. So within the nuclei, you see the chromosome. Now, within the chromosome, how is the DNA being wrapped around? Okay, so this is, once again, you would see it in multiple textbook, but we'll be talking a little bit more about whatever is there in the textbook. So within the chromosome, if you further unwind, the DNA is actually tightly wrapped around. And then if you further unwind, then you start from chromosome to a supercoil structure. And from the coil structure, then if you further unwrap it, then what you see is that it's what we call the 30 nanometer fiber. Now, these 30 nanometer fiber is composed of, the basic unit of is the nucleosome. Okay. Now, the nucleosome contains the DNA. So the entire DNA is actually tightly packed around within the chromosome, and so that the DNA, as you can see, every DNA, or rather what we say that 147 base pair of DNA is tightly packed with these proteins called histone, which 8 of these histones come together to form a nucleosome that further forms the 30 nanometer fiber, and this 30 nanometer fiber then further supercoils into a structure to give rise to the chromosome. So as you can see, that the DNA within these chromosomes, it's because it's so tightly packed around, it's not accessible, readily accessible for the purpose of the gene expression. Okay, and because of that, the default state of any given gene for their expression is to remain silent, because it's easy, because it's tightly packed. So the genes do not get expressed. That's the default state. So depending on what you need, then you turn it on. And that turning on processes, what we called is the gene expression. Okay, so when we go for the gene expression, and this is a basic central dogma of the molecular biology, and I'm saying all these for some of the diverse other people who are there who may not be so well accustomed to this molecular biology, but for those who have already read it, please bear with me for a few slides. I'll go with this to bring everyone on board with the topic that we'll be discussing. So this is the central dogma for the molecular biology in terms of gene expression. So the DNA that we talked about, from the chromosome to the supercoiled structure to the 30 nanometer fiber to the nucleosome, and then you get the DNA. So from the DNA, and we get, by a process within the cellular system called transcription, we get the RNA. From the RNA, by another process, we get the protein, and that from DNA to RNA, the process by which we get the RNA, it's called transcription. And from RNA to proteins, by the process by which we get the RNA to protein, it's called the translation. Now remember, it's the protein that's actually functioning within our system. Everything is being regulated by the proteins that are there within our system. So it starts from DNA, which encodes a specific type of RNA, from RNA that ultimately give rise to the protein by the process of translation. So what we are going to talk about the gene expression today is the process of transcription, which is shown over here in green color. Okay, and how we can regulate the process of transcription for the expression of the protein, and then subsequent different types of diseases and all others that we'll be talking about today. Okay. Now remember, for the cells to or any given organism to perform or to do properly function and to regulate everything, so it has to have a proper cell cycle regulation, because within our system, cells are always dividing. Cells are always dividing, and then when they divide from one cell to another, what we call the cell division. Now, during this process of the cell division, you have multiple stages, what we call as the G1 to S to G2, and then finally M for the mitosis phase. Okay, so different stages are there. In the G1 phase, cells acquire everything. In the S phase, from one DNA, you get the 2 DNA composition. Then the DNA has to be further divided to make so that the DNA gets carried to the next generation. So that during the cell division, we get from one parental cells to 2 daughter cells. Okay. Now these cell divisions are always important for the biological functions, because within our system, cells are always multiplying and the cells are always degraded. So that old cells have to be replaced with the new cells, and the new cells come through this process of the cell division. Now remember, while doing the cell division, the process of the cell division, you have to have a perfect control of this cell division. Otherwise, there is a problem. Is that whenever, let's say, the S phase, which is shown over here as a DNA synthesis, okay? Now, if there is a problem with any phase, any stages of these processes, then there is a checkpoint, what we call the cell division checkpoint. Okay, as you can see, it's written over here in this red highlight. The G2 checkpoint is there, G1 checkpoint is there. So cells always kind of monitor through this checkpoint in terms of whether everything is fine or not. Okay, and then only it allows it to go into the M phase or the mitosis phase to make to the 2 daughter cells. Okay, now if there is a problem with this checkpoint, then what happens? Cells really do not recognize that if there is any problem, anything, it continuously proliferates. It doesn't respond to the external cues. It doesn't respond to the other signals saying that now you will have to stop. And that causes different types of problems, all sorts of problems, including the gene expression related all the diseases and cancers. Virtually a lot of diseases that we are going to talk about. Okay. So a proper cell division from one cell to other with specific checkpoints are absolutely important. And these checkpoints are the proteins, and that expression of those proteins are tightly regulated. If there is any problem with the expression of this protein, then there is a problem with the cell division and ultimately giving rise to the abnormal cell proliferation, which starts with a tumor formation, giving you the cancer, starts with, or the reduced expression, or the cells do not divide, and then reduced expression of the proteins, ultimately giving rise to different kinds of diseases. Now, this is important for the proper function. And as I said, and in these cartoon diagram, it is that in a normal cell, you will get, depending on the growth factors. If there is a good growth factor, you are feeding every day and all these, and so the normal cells will divide from 1 cell, 2 cells to 4 cells, 4 cells to 8 cells. If there is no growth factor, the cells do not divide. Okay. However, in the cancer cell, what happens? As I said, that their checkpoints do not function. So the cancer cell, what happens is that they become immune to the with or without the growth factor. So whatever is there, they will keep on proliferating. And because of that, then all sorts of problems happen, including generation of tumors as well as cancers, and then ultimately leading to all these types of diseases. Now, this is one of those in the right panel is shown how many different types of ways that cancer cell can give rise to. And you can see it's a multiple ways, including different types of cell cycle regulator, including other external cues that becomes a problem, and all these. So it all depends on ultimately with the proper expression of all the factors, gene expression of all the factors, ultimately lead to the proper functions of our human body. Okay. Now, what happens is that if a cell abnormally and becomes kind of refractory to the external cues, and also become kind of they cannot become sensitized, cannot it's not as sensitive to the external cues or other signals. What happens is that these cells keep on dividing. Okay, and also because they out proliferate the other normal cells in terms of their overall proliferation ability. After a period of cell cycle, after a period of growth stage, they become really prominent, and then ultimately those cells become really kind of dominating one. And because of which, whenever a cell acquires any kind of ability to have the properties of cancer, it outnumbers the normal cell, and ultimately that those are the cells that grow, and the normal cells then ultimately become outnumbered. And because of which the cancer cell, because they can grow even with or without signals and all these things, so their fitness is very high, as opposed to the normal cells. Normal cells will demand that I need these, I need these, I need that. However, the cancer cells, they become resistant in all this, and then they keep on growing, and then at some point their fitness becomes so high that they outnumber the growth of the normal cell. Now, during these growth of the normal cell, of the cancer cell as well, not the normal cell, in this case, let's say cancer cell, you need to have a continuous production of the proteins, because all other processes, from cell division and all those things, you need to have those proteins. So the cancer cells also need to produce the protein like the way normally it's being produced in the normal cells, but they become refractory to some other signals, and because of which they keep on growing. Okay, so the overall processes of, if we know how the process from a DNA to RNA to proteins is being formed, okay, and if we know that how these processes are being regulated, and further, if we know what are those proteins that the cancer cells rely on, then we can think about some therapeutic strategies, some intervention strategy for the purpose of mitigating those diseases, which is exactly what we are going to talk about, but restricting ourselves to the process of transcription, from which means from DNA to RNA. Okay. Now, whenever the process of transcription happens, and some of you might already be knowing, but then I'm giving you kind of a recap of these, because to bring everyone on board. So this is a cartoon diagram of a gene structure within mammalian cell. Okay. Now, these gene structure, okay, and it starts from a promoter region, okay, and then from promoter region it has two parts, so promoter region and the coding region. So in the promoter region, then all the transcription machinery would first assemble on the promoter region, okay, and by two steps called the pre-initiation and the initiation, and I would go a little bit deeper into it. And as you would see, I'm going a little bit more deeper into the overall processes to make you understand that where we could be, we could actually target these overall processes. Okay, so it starts with a pre-initiation, then goes into the initiation. So in this initiation means it just is the beginning of the starting of the process of transcription. So all other factors come and then they assemble and then they start the process of or initiate the process of transcription. Then it goes into the next step of this transcription called the elongation. So once it initiates, then it has to elongate. It goes into the elongation phase within the coding region, and when it reaches to the end of this gene, then the process of transcription has to end, which we call the termination. Means it's going to terminate the process of transcription, and thus making an mRNA. In this case, what we are going to talk about an RNA or rather mRNA, because we're going to mainly focus on the RNA Pol2 rather than Pol1 or Pol3. Okay, so that you get a functional mRNA, which would be next be translated to give you the protein. So what happens to the process of transcription? As I said, the basic, the default pathway of any given gene is to remain silent. So you have to, whenever you need to turn any given gene on, then you will have to really spend some energy to make the DNA available, because DNA otherwise it's tightly wrapped around. Unlike the bacterial DNA, which is more loosely structured as opposed to the eukaryotes or the mammalian, which is what we belong to, their DNA is really, really tightly wrapped around. And so DNA is system. Okay, so there are cellular machineries which actually helps in making the DNA accessible, which we call the chromatin remodeler, which remodels the chromatin, and then that helps in making the DNA accessible. So once the DNA becomes accessible, then what happens is that, and one of these, as I said, the initiation, the process of transcription starts with the pre-initiation, then initiation, elongation, termination. So during the process of initiation itself, there are multiple factors which gets recruited at the promoter region. Okay, and the goal of all these factors at the promoter region is to bring in or recruit the main machinery which actually transcribes the underlying DNA sequence from DNA to RNA, is a protein called Rna Polymerase. From the name itself, you can suggest that it's a polymerase which makes the RNA, which we are going to talk about is that RNA polymerase 2. So multiple other transcription factors, okay, including the ATP dependent, and this cartoon diagram shows the including ATP dependent chromatin remodeler. The name itself is a remodeling, means it's making the chromatin in such a way so that the underlying DNA sequence become available. Okay, and then the other factors come in, like histone modifying enzymes, which I will be talking about it a little bit later. Some other initiation factor called the mediator or some other cofactors, and all these things ultimately culminate into the recruitment of the Pol2, RNA polymerase 2, which we call it as a Pol2, which then once it gets recruited, then by the action of some of these factors, which says the RNA polymerase 2, okay, now that you have got recruited, now it's time for you to move. So RNA polymerase then starts from that promoter region to going into, and it starts from a specific region, basis of the within the gene, which is called the TSS or the transcription start site. So for every gene, there is a defined TSS is there. So whenever the RNA polymerase 2 is getting recruited, then it will start transcribing from the TSS and it will go into the encoding region to make the functional RNA. So it will go into the coding region, then the job of the initiation factors are over. Then the elongation factors. So for every step, there are defined factors which would do at the which would be involved in regulating these transcription processes depending on where RNA polymerase 2 is. So the job of these initiation factors is to recruit the RNA polymerase 2. And then once it starts, then the job of the elongation factor begins. So it will help in elongating the RNA polymerase 2 to elongate into the coding region. Okay. And then it will go slowly into the, depending on if it's a big gene, it will take a longer time. Then if it's a shorter gene, then it will take a less time to reach to the end of the gene, and then wherein the process of transcription ends by the factors, once again, dedicated factors called the termination factors, which would help in which would assist in terminating the overall processes. Okay. Now, this is the kind of cartoon structure of RNA Pol2, which you can think about, wherein, as you can see, the RNA Pol2 or polymerase 2, when it goes into the DNA, then it makes this, it has this, a channel-like structure, wherein it, with its help of the unwinding help of these functions within the RNA Pol2, which has a 13 subunit, which helps in unwinding, and then it has this catalytic activity wherein it helps in making the phosphodiester bond with the incoming RNTP, and then it keeps on adding that, and then the RNA comes through the exit channel, and that's how it goes into the overall process of transcription. Now, you might be thinking, if this is how it is, so how efficient the process is. You would be surprised to know that the processivity rate of RNA Pol2 within our cellular system, it's about 2 kb per minute. Means RNA polymerase 2 is transcribing almost 2 kb per minute. So it's a fairly efficient machinery which helps in making the RNA from a DNA structure that is there within our system, within the nuclei or the chromosome. Okay, from there you get the underlying DNA sequence, and then RNA Pol2 starts moving, and during that processes, all these transcription factors come in and help in these processes of regulation of the transcription to make the mRNA, which would further be utilized by the process of translation to make the protein. Okay. Now, how do we see that? So this is one of those examples. I've just given you one of this data, over here to give you an example that how we actually see the process of transcription. So if you take any given DNA, okay, and then if you add the RNA Pol2 into it, in this panel C, which marked as panel C over here, in the control, if you start the process of transcription and let the RNA Pol2 go, so it will keep on going by virtue of its own activity. It will keep on going to make the RNA, as you can see, from the len number 2 to len number 7. So more time you give, more longer RNA that you see that's being formed. Okay. Now, in this, if we use the elongation factor, okay, which is marked over here as ELL.com, which is a typical elongation factor, then what you see that if you compare the overall production of the RNA in the ELL.com, you see at 2 minutes time point it reaches almost to the end. Whereas the control reactions, you don't see that much of the elongation of RNA polymerase to give you the RNA. So that's how we actually study the overall function. This is a typical in vitro studies wherein we add the DNA, we add the purified proteins and everything. In this case, we have taken the DNA, we have added the purified RNA polymerase 2 to let it go, and then it starts transcribing. And then if you add the elongation factor, then it makes it faster, because as you see at 2 minutes time point, in the len number 2, you see it has reached to the end, whereas in the control in len number 3, it's still going slowly. Okay. Now I said that, as I said, and some of you might have heard about this, as I said, that the overall regulation of this gene expression, okay, it's not just the DNA, it's not just the histone. However, there are other components of it. So the histones that we are talking about, there are 2 copies of all this histone, histone H2A, H2B, H3, H4. These are called the histone octamers, and that wraps around the DNA within this chromatin structure and to form a kind of supercoiled structure to give you the DNA, to give you the nucleosome, chromosome, which is what you see within the nucleus. Now these histones, which actually wrap around, and this crystal, this is a crystal structure of histone H3 is shown over here. Okay, when you cut it into half, okay, in the right panel. Now within these histones, what happens is that histones, it has a two kind of structural component. One is a core And another one from the core, it has this tail structure, which you call as a histone tail. Okay. Now the core structure and the tail structure ultimately forms the histone. Okay. Now, interestingly, this tail structure, which is protruding out of this nucleosome structure, which is shown over here in these, a kind of a bubble-like structure. Okay. The tails protrude out of this. Okay. And these tail structure of these histones are kind of, you can say, a launch pad, in a sense that these tail structure of the histones, they get modified for the regulation of the gene expression. What kind of modification that we see? We see multiple types of modification, including acetylation, including phosphorylation, including methylation, multiple others. And one of those examples of histone H3 and the tail modifications are shown over here. As you can see, phosphorylation, acetylation, methylation, and so many modifications that could happen in the tail as well as in the core structure. Okay. However, tails are kind of protruding out. If this is the globular structure, tails are protruding out of it, and there's a readily accessible. Okay, and that gets, depending on whatever the outcome of the gene would needed be, then tail gets modified accordingly. So you can think about any given combination of the acetylation and a phosphorylation can give a different output than a methylation and a phosphorylation, or depending on which residue you are talking about. Let's say if you are talking about, in this case, histone methylation at H3K4, this is histone H3. H3K4 methylation is associated with the transcriptional activation, whereas same histone methylation at H3K9 gives you the transcriptional repression. And how does that happen? Okay, I'm giving you a little bit of these so that afterwards, whenever we'll be talking about the CRISPR-Cas9 mediated, mitigation of this, it will be easier to understand. Okay. Now, these tail structure, which gets modified, as it's shown over here, they ultimately, these tail structure regulate the recruitment of multiple factors for the processes of transcription, for the processes of regulation of transcription. So the overall regulation for the process, as I said, any given gene, the default state is to remain silent. And then when you need to activate it, then you put the efforts and the energy to activate it. Otherwise, because overactivation, overexpression, or a lowering expression is always a problem. So that happens with, let's say in this case, with a tail modification. And these tails now would be recognized by multiple other proteins. Okay, so those who are writing the tail, all this, giving you the acetylation, okay, or a methylation or a phosphorylation, which we call by the histone code hypothesis, these are the writers of this epigenetic modification, or in this histone modifications. Okay, so the writers are writing depending on if you all of a sudden, if you are under stress, or all of a sudden if you are undergoing kind of watching a horror movie or something, so a specific set of genes have to be expressed, or some other scenario. So in that case, a specific writer then goes and, depending on the cues, and writes something on the histone tails. And that gives you either a combination of acetylation, phosphorylation, methylation, depending on whatever it is. And then once the job of the writer is done, then comes the reader. Okay, the reader then reads these modifications. So the readers have a specific protein or specific domain which can recognize a specific combination of these modifications. And thus, whenever there is a specific combination, the readers will then read it. And that would help in recruitment of the transcription factors for initiating the process of transcription. Let's say the process of, let's say, a histone H3 lysine 4 reader, MLL complex or set1 complex, that writes histone H3 lysine 4 methylation. That gets recognized by one of these transcription initiation factor called the TF2D, and that helps in recruiting TF2D. Okay, and TF2D, or the initiation factor, then would recruit the other initiation factors, and ultimately leading to the recruitment of the RNA Pol2 for initiating the transcription. Now, once you are done with the expression, because you cannot have those genes which are getting expressed, always turned on, because that's a futile, right? So then you will have to bring it back to the normal. So that normal function is being done by other proteins called the eraser, okay, which erases the whole writing and brings it back to normal. So that afterwards, whenever you need it again, then you'll have to restart the process again. Okay, but if it happens to be that the same genes have to be turned on within a very short period of time once again, then it becomes relatively easier to turn it on, because some of those modifications would be there and eraser still could not erase the whole thing. Okay. Now, what are those writers, erasers, and all those things? So depending on which specific histone residue that you are talking about, and over here, some of the H3 tail lysine residues are shown over here. Okay, as you can see, the scientific literature or scientific literature has found out so many of these writers as well as erasers. So it all depends on which cell line, which cell type that you are talking, which context that you are talking. Okay, the same type of cells with a different context would be having a different kind of writer than a different or a whole new different type of context. Same way, the erasers would also be different, depending on the cell type, depending on the specific context. So you have a combination of writers, erasers, as well as readers to ultimately regulate the process of recruitment of all these factors onto the promoter region, let's say, to start the transcription. And as and when it goes into the coding region, there also you have these histone code hypothesis for regulating the expression of the gene. Over there also it works, but a different type. At the initiation, you have the histone H3K4 or the lysine 4 methylation, whereas in the elongation, you would see more of histone H3 lysine 36 methylation and histone H3 lysine 79 methylation. Okay, so depending on where you are talking, you get different types of modification that ultimately helps in recruiting all the either initiation factor or elongation factor or a termination factor, ultimately regulation of this entire processes. Okay, now as I say, this whole erasers are different types of, and as an example of the lysine acetylation or the histone acetylation, which being written by this protein complexes called the HAT or the CAT. HAT was the earlier name for the histone acetyl transferases, which helps in transferring the acetyl moiety to the histone lysine residues, whereas the reverse reaction is being done by HDAC or the histone deacetylases. Nowadays, this has been changed to CAT or the lysine because these HATs have been found out to not just regulate the acetylation of the histones, but also the other non-histone proteins as well. Okay. Now, as I said, the histone code, and I'll not go into the details of it because I talked a little bit about it. So depending on what type of cells that you are talking, depending on what type of context that you are talking, so you have a code, specific code. Now, however, let's say histone lysine 3K4 methylation would do the same job irrespective of the whatever cells that you are talking about. So that is fixed. But whatever the overall combination of writing that you would get and ultimately leading to the outcome, that depend on the cell type to cell type. Okay. Now, once again, all these would ultimately lead to the recruitment of the factors with the histone code hypothesis, with the underlying DNA sequences, whatever is there. The DNA also gets modified by methylation by the enzymes called the DNMTs, which silences the genes. So all this ultimately helps in recruiting these factors, the transcription factors. And it all starts with first recruiting the activator. Okay. So by name itself, as you can see, it helps in activating the transcription. So the activator first gets recruited, and that in turn recruits in other, aided by all these histone modifications and all other signals, in turn helps in recruiting other transcription factors, in this case initiation factors, to help in ultimately finally recruiting the RNA polymerase 2, which initially, with the help of some of this initiation factor, it escapes from the promoter, which we call the promoter escaping. And then once it goes into the coding region, then the elongation factor takes over, and this it helps in further enhancing the overall rate of the rate by which RNA Pol 2 transcribes and then helps in overall transcription processes. Okay. Now, in this process, there is another element to it for the purpose of this whole regulation, which you call the enhancer. Okay, the enhancer region, as by name itself, it suggests that it enhances, means it increases the overall transcription. Now, this enhancer is not part of this whole, the gene that you define with the promoter and then coding region and at the end. The enhancer, it actually lies sometimes several 100 kilobases away. Okay, but that 100 kilobases away, it happens when you see the DNA as a linear stretch. But when the DNA is there within the chromatin, within the chromosome, that's not in a linearly, packed into, because it's in a 3D. What happens? Some of these enhancers, what happens is that although by linear fashion they are hundreds of kilobases away, but in a 3D they come close to the gene that they will be regulating, and enhancers always keep the gene on. Enhancers, the overall transcription within this enhancer always remains on. And as and when you have to turn on any given gene within a very short period of time, let's say in a developmental stages, okay, when a lot of things are happening, you have a very short any given gene. So because it just keeps on the transcription within its enhancer boundary always, and then as and when with some external cues, it needs to pass the message on, then the enhancer passes the message on to the gene, and then that goes in. So the enhancer actually helps in passing on all the transcribe, call to, and other machineries to the gene sometime to help in processes of transcription. So it helps in enhancing the process of transcription, because I'm bringing this up because in few of those contexts that we'll be talking about the modulation of enhancer for transactivating the target gene expression. Okay. So now I would go a little bit into, this is kind of what I gave you, the overall overview of the transcriptional regulation and how, within the mammalian cells, how we can think about the overall process of transcription, and then from the chromosome to the supercoil structure to the 30 nanometer fiber to the nucleosome histone to the DNA, and then how multiple factors assemble onto a given promoter and ultimately leading to the expression of the genes. And all these are tightly regulated, because otherwise, if those are not properly expressed, then you have a problem, and we don't want that. Okay, so that is the underlying principle by which we who are working in the field of gene expression, we look at the disease aspects of it. Of course, there are other parameters also which ultimately regulate the outcome of the pathogenesis of any given diseases. Okay, but in terms of gene expression, whether how the gene expression is being kind of misregulated, either at the initiation or at the elongation or at the termination stage, and ultimately leading to the aberrant cell cycle, regulation of the cell cycle, and leading to the proliferation of the cells and all other diseases that we are talking about. Okay. Now, how we can actually use the CRISPR technology for understanding some of those diseases as well as their mitigation. So I will go slowly one by one, some of those examples that I have compiled for this presentation. And let's first talk about one of the basic things that we do is to use this CRISPR technology for screening the cancer-associated genes. So the overall idea is that the gene expression, so the cancer cells, okay, cancer cells, as I say, would need the processes of gene expression for making the proteins for its own proliferation. Okay, because when it's making from one cell to another, it cannot have or it cannot survive to have a faulty DNA or something. Okay, although it does not respond to the checkpoints and all those things, those are different, but it needs to have the basic machinery always functioning. So for that, it needs to have the gene expression always on. So if we know what are those factors or how the cancer cells are being dependent on the expression of any given gene, so that we can afterwards target those set of or those gene or those specific gene or set of genes for the purpose of mitigation of this disease. So, this is the basic of this CRISPR, as you may have already heard from the previous two speakers, is that how it works is that primarily there is a gRNA component of it and the Cas9 component of it. gRNA part, it recognizes the cellular DNA, which is there within the chromatin, as we have talked about, and there is the PAM sequence, and there's a nuclease called the Cas9, which cleaves close to the PAM sequence, and that gets the DNA when it gets cut. And I'm not going to go into this because there are other speakers who would go to more details into these overall processes. So I'm going to just give you a brief idea of it so that we know that how we are actually going, how we are going to kind of target it. Okay. So whenever it makes a cut, then it gets repaired by two different processes. One is called the non-homologous end joining, another is called the homologous recombination. Majority of the time, the cells employ the NHEJ or non-homologous end joining, which is a faulty machinery for the repair, and it gives rise the mutation within these at the cut DNA when it gets repaired, and then that ultimately inactivates the whole gene. However, also at the same time, the same processes, can be faithfully repaired by the directed approaches by the homologous recombination to make or edit specific type of sequences we would like to. So we can do this kind of manipulation always for proper control of the gene expression. Okay, let's go some of these examples. So the cancer one that I have talked about earlier. So in this case, usually what they do, and multiple other components have already been targeted in the literature, and then some of those are shown over here. So we would just show one or two of those components in terms of overall understanding on how people are thinking of using this CRISPR technology to mitigate the disease of cancer. Okay, so now one of these very commonly used in our experimental system is called the screening of CRISPR-based screening in cancer. So in this case, what happens is that you make a library of the gRNA, which would actually target pretty much most of the genes within the eukaryotic cells. So you can make this library. So the library represents all the gRNA targeting all the 20,000 genes which are important for the mammalian cells. Now these, you make this pool of library, then you take the cancer cell and then you infect. You infect the cells with a lentiviral. What usually we use the virus-mediated delivery of the gRNA. Okay, and then once you put this into the virus-mediated delivery within the cancer cell, then you see how those cells are responding. You can either use the mouse model or just an in vitro cellular based. The top two panels A and B are shown for the mouse model, and then the bottom panels are shown for the cancer cells in vitro, and then you see the proliferation of it. Okay, how the cells are responding to the overall knockdown of the, in this case, main idea is to do a knockdown, the gRNA mediated disruption of the cells in an unbiased way. So because your pool of library is representing the overall 20,000 genes, so you are going for unbiased. So once you get cells which are out proliferating the others, and then you can see that then you can use the conventional because all these gRNA can be ultimately tracked because it has a barcoding in it. So we can always track it. So once we see the outnumbered cells, then we can barcode it, or the cells which are proliferating less, whatever your criteria is, we can then sequence it, and then we can say that, okay, because of presence of this gRNA or the disruption of these gene, the cells possibly, in this case, I'm saying possibly because this is just the beginning of the entire study. Okay, these gives you the overall hit or the lead targets that would actually ultimately tell you, okay, you can further study based on these screening. You see these genes, those could be targeted, and then you subsequently target those genes, and then you choose few of those for the downstream in detail study to show the role of those specific genes for the overall cancers, depending on the type of way that someone would like to. Okay. Now, one of the biggest type of gRNA-based cancer cells and screening of the cancer cells, proliferation and all this have been done and have been published in Nature just a few years ago. And what they did is that they took almost 324 cancer cell line, representing 30 types of cancer. And then the total gRNA represented around 18,000 genes. Okay, with these, whatever that they had, and I'm not going to go into the details of all this, that's beyond the scope of this lecture that I'm giving you, but all what I would like to say, with once you get all the NGS data and all these things suggesting that, okay, this gene is required, this gene is required, then you fit it into a algorithm is there, and that algorithm gives you that, okay, for this specific type of cancer, for this specific type of cell, then this cell is depending on the expression of this set of genes for their own survival. If you have any kind of disruption, then that cells cannot survive, and then so you can actually then target those specific set of genes. Okay, and one of those examples are shown over here in panel B and panel A, B, and C both, and all the genes that are shown in panel A, and those are all color coded. Okay, and a specific type of cancer, which is shown in the bottom panel of the panel C, and specific type of cancer, its dependability on expression of these genes, whichever is shown in panel A, and how much, and their overall scores. Everything can be done to give you the overall idea that how multiple types of genes, ultimately their overall expression or the overexpression, ultimately co-regulation, lead to this, giving rise to this type of cancer. Okay, so and you do the further analysis, further all those by the cell-based assay, by the mouse model, by others, to ultimately show that, okay, these are the specific set of genes. So you start with a CRISPR library to find out the targets, and then you go in details for selecting few of those to show, okay, these are the critical genes, those are required for giving rise to this kind of cancer. That can further be targeted for, for a small molecule-based inhibitors or others for mitigating those diseases. Okay, so now this is about the deletion of the CRISPR that I say. Now, a couple of other slides that I'll be showing. And in this case, what we are actually trying to get or trying to modulate the expression, the one that I talked about CRISPR library, that's talk about the genome-wide. But the one that I'll be talking for the next few slides is that we are going to turn on a specific gene within the mammalian cells to see the effect of it. Okay, so in this case, the one of these concepts that's become very critical is called the concept of the dCas9. Okay, I don't know if some of the earlier speakers may have talked about it. If not, the idea is that the dCas9 or a dead Cas9. Okay, in this case, it's catalytically dead. So the Cas9, which is supposed to cut after it gets sensed by the gRNA, supposed to cut, it doesn't cut. So what happens is that now the gRNA, it goes there and it annuls to the specific type of cells, a specific type of DNA sequence. Okay, now if you express the Cas9, then it goes and binds over there in that at that specific locus, specific gene. Right? Now, if you use anything with the Cas9, dead Cas9, because this Cas9 is the dead, it's not going to make a cleave or anything. It's just it will get and it will go there and it will be recruited. Now, if you fuse this Cas9 with other modulator, let's say in this case transcriptional activator, which we'll be talking about, a histone acetylase, which usually it's involved in transcriptional activation, a histone deacetylase, which would be involved in transcriptional repression, a histone methyltransferase or a histone demethylase, or other ways of regulation. Then you can think about that by expressing a specific set of gRNA, and then you can go and target a specific gene. This is even more important because a lot of time we see that the cancer cells are being dependent on cancer cell, not just the cancer cell, even other type of disease pathogenesis is that some of these genes are either overexpressed or underexpressed. So the genes in which you need those which are underexpressed, you can always target those genes by the dCas9. You just express some of this gRNA, that gRNA would go and then bind, and then you use a dCas9 or a dead Cas9 would go and bind that, and if you fuse with an activator or a histone acetyltransferase, and that will activate the gene. the genes which are silent, and because of which the cells were having the proliferation advantage, now it will not be having proliferation advantage, or the other way. So you can think about using this Cas9 and expressing a specific set of the gRNA, then you can target virtually, I'm saying virtually and literally, kind of any gene. But there are some limitations which we'll be talking about it afterwards. The reason that people got excited about it, particularly because of these abilities that wherein you can target a specific set of genes, or a specific gene or specific set of genes for their either activation or a repression. Okay. Now, so we'll be talking about this modulating the expression. Okay. Now, as I said, once again, this slide I have shown you before. So the histone can get the acetylated. Acetylation is for the transcriptional activation, whereas the methylation, it depends on depending on whichever residues. If it is a histone H3K4 methylation, then it activates the transcription, whereas the K9 methylation, which is being done by this eraser by the writer, then you can repress, or you can turn the gene, you can turn off the gene, the expression of the genes. Okay, so giving you one of those examples. Okay, so in this case, and this paper was published in Nature Biotechnology just a few years ago. In this case, what they have used is that they have used as shown in this panel A, they have used the Cas9 or a Cas9. Okay, and they have used multiple types of activator, transcriptional activator. Remember the activator that we talked about earlier? So they have used one activator called the BP64. Okay, they have also used for activating a specific target. They have also used one of these acetyltransferase, or very potent acetyltransferase, is called the P300, or a histone acetyltransferase, you can call it as well. P300. They have also used, because P300 is big and there are some challenges to it, they have also used just the acetyltransferase domain and in the shorter form of it. Okay. Now, when they have used these in combination with the gRNA and expressed it within the mammalian cell, what they have used when they have used the gRNA for the IL-1, or the genes, those are shown in panel B, they can see that as compared to the control, you can see, as compared to the control, when they have used the P300 code, the target gene expression went significantly up. So you actually can go for specifically targeting a specific gene by fusing it with the activator or other histone modifiers, or turning the gene on of that specific target. Okay. And then some of the others are shown over here as well, in which case they have used multiple other gRNA with a MyoD, OCT4HB, and other target specific targets for turning on the gene. And these are very specific, because if you use the other gRNA, if you use, if you look at the expression of the other genes, those do not get transcribed, do not get upregulated, but a specific set of genes. Okay. Now this is about activating gene by fusing with the activator, and you can always repress it. So as I said, the histone, when histone gets acetylated by histone acetyltransferase or lysine acetyltransferase, same way it gets deacetylated by deacetylase for bringing it back to normal. So deacetylase is usually involved in if the hat is involved in activation, the deacetylase is involved in transcriptional repression. So they have used, and this paper was published in 2017 in Nature Communications, wherein what they used is that they used, once again, the Cas9, as I said before, in this case, the Nic deficient Cas9. They have fused the Nic deficient Cas9 with the HDAC3 for the transcriptional repression. And in the panel B, it's shown is the normal level of expression of all these genes. And then when they use different types of gRNA, as shown in the position as shown in this panel A, and then when they do the next generation sequencing for seeing the overall level of the activation mark and the repression mark, what they see, the histone H3K27 acetylation mark is significantly down in the case where they have targeted for the, in this case, they have targeted the ABCP2 gene. And if you see in the enhanced part, that the histone acetylation is significantly down at specific locus that they have targeted, as well as their expression. And this is very specific to the functions of the HDAC, because when they have used the HDAC3 deficient construct, they could not, the H3K27 as shown in panel C, H3K27 acetylation mark, which is an activation mark, which did not go down. So suggesting that this is very specific to that HDAC3 being recruited over there. And then overall level of the HDAC3 and the acetylation goes down, as well as shown in panel E in two of these red color box, is that leading to the transcriptional downregulation of the MECP2 gene that they have targeted. But in that paper, they also gave numerous examples of other non-targets showing that this overall approach is very effective. Okay. Now, this is about the overall, I talked about the histone acetylation and the histone deacetylation. Now, other transcription repression or transcriptional negative regulation of transcription, or the what we call the repressor proteins of transcription, that can also be targeted, not just the histone deacetylase, but other, by virtue of them, these transcription factors are the repressor, that they do interfere with the Rna Pol2 mediated transcription, because that is also important for bringing the overall function to normal. So in this case, what they have used another factor, the Crab domain containing factor called MECP2, and this is typically involved in transcriptional repression. What they have used this Crab MECP2 with the gRNA expression, the dCas9 gRNA, and targeting a specific locus. In this case, initially they used the luciferase reporter, and as you can see in the panel C, the expression wherein these factors are being expressed, the expression of all these genes are going down, whereas they have done multiple other experiments with the native target genes, including the reporters, what I have told you earlier. And as shown in panel C, when they use this Crab domain containing, which is fused to the dCas9, then you can, along with the gRNA, you can target a specific gene, in this case KMT2B, RELN, and NPY. It's shown over here. Okay, so with these, you can actually then either depending on what you fuse. If you fuse an activator with the dCas9, then you will turn on the gene. If you fuse a repressor with the dCas9, then you will turn off the gene. So you can actually modulate the expression of the genes. Now, remember the CRISPR-Cas that we talked about, the library. So from that, if you know that in any given cells, these are the targets in which these genes are going up, these genes are going down, and the overall modulation or overall survival of the cells depend on whether this set of genes go up versus this set of genes going down. You can actually then modulate these overall expression. If they're going up, then you turn it back off. If they're going down, then you turn it back on. Obviously, it's not all of them equal, but you'll have to be selective. So depending on that, you can actually modulate the expression to look into the overall outcome in the overall cancer pathogenesis or even other diseases. Okay, as we talked about, so enhancer, as I talked about earlier, that that's residing long further away, but then in the 3D they come close to the gene. So the enhancer proteins, sometimes we also can regulate the functions of the enhancers as well. Okay, so that these enhancers become not so much active, or depending on, we can also turn on the enhancers to become more active, to express, to increase the expression or reduce the expression. So one of these examples are shown over here, and this paper was published in Nature Genetics just I believe about 9-10 years ago, in which they actually targeted the enhancer region for modulating the expression, or in this case for target enhancing. And this because they have used a VP64 activator with the gRNA, and multiple gRNA also you could use for turning on the enhancer, so that when the enhancer gets turned on, the overall ultimately regulating, turning on the target genes in which enhancers are ultimately regulating the expression of. Okay, in this case, they have shown the MyoD as an example, and they have used the various either acetyltransferase or the activator to show that the genes can be turned on by targeting the enhancer region. Also, they have used some other targets as well. Okay, and along with it, they have shown that the multiple other enhancers or multiple other genes that they have targeted can be targeted for turning on the genes, so that you can modulate the expression. Okay, and some of these are shown over here. Okay, Now, this is about the modulation, the targeting activation. Now, you can actually also make it. They have also shown in that same study. You can also make it as a super enhancer. Means an enhancer which has, let's say, a function of, if you put it a categorized, let's say it has a function of, let's say, 40%. You can make it to even, let's say, 80% by adding some more component within the enhancer, so that it becomes a super enhancer and a zombie kind of thing, so that it activates. And as shown over here, that they have targeted, they have inserted within the enhancer sequences, they have inserted another activator sequence called the MIB sequence, which actually makes this gene express. This is an oncogene which expresses even more and causes us even more proliferation. And this is why you see in the lower panel and panel B, when they use the SG-MU2, then it becomes even more proliferative, it becomes even more metastatic, and that's why the red signal, because the red signal represents the metastatic property, the red signals are more in the mouse as compared to the control GAL4. As you can see, you cannot see any red signal in the control GAL4, suggesting that you can also modulate the enhancer to make it even further. So depending on whatever you need, either more or less or whatever, because you can also target the overall, you can also modulate the overall level of this enhancer molecule to be targeted. So depending, and ultimately giving rise to the precise control. Okay. Now, this is, I talked about the enhancer activation. Same way, the enhancer can also be down-regulated. And this is one of those wherein, once again, they have used the CRAB domain containing the repressor protein, in this case D-methylase, called the LSD1, okay, which is a histone demethylase. And when they fuse it with the dCas9, as you can see, some of those examples are shown over here, the overall expression of all these, and relative mRNA expression of HB1 goes down, so are the others, okay? So you can regulate the enhancer function, you can regulate the gene wherever, either at the promoter region or somewhere at the coding region, to modulate the overall expression of the, but in terms of regulating the gene, it's always preferred to start from the promoter, not at the coding region, because that way you get precise regulation. Now, once again, is another example of down-regulating the expression of the genes, wherein you fuse the dCas9 with another repressor protein called the EZH2, which is a histone H3 K27 methyltransferase, which is involved in transcriptional silencing. So you can once again see, as you can see, when you express this dCas9 with the EZH2 in panel C, the overall level of the histone H3 K27 methylation goes up, histone H3 K9 methylation goes up, which are all the repressor marks. So suggesting you can actually target these histone modifier for reading or writing a specific code for regulation of expression of the genes. Okay, so I'm not going to go into this. Let's just skip some of these because time is almost running out. Okay, now another one. What we talked about, the histones. Now let's talk about some of these proteins. Okay, proteins means, in a sense, that the expression of those genes also can be targeted. So in this case, one of these very common genes which are involved in the breast cancer is the BRCA1, which is involved in the DNA repair. Okay, so what happens is that the breast cancer gene, the BRCA1 gene, if there is a mutation, it doesn't produce or it doesn't express because of the mutation within the gene, okay, because of several hits and all these things. So the BRCA1 doesn't express, and then you have a problem of the repair and ultimately giving rise to the breast cancer. Now, these repression mechanism is being done by the DNA. In this case, we are not talking about the histone methylation, but rather a DNA methylation, which represses or which turns off the gene. Okay, so the BRCA1 doesn't express. Now, these histone methylation can be once again reversibly demethylated by one of these enzymes called the AICTE, which demethylates is DNA methylation, whatever is there. Mainly it's there in the CpG island, and demethylates and activates the gene. So what they have shown that if you take a TATE1CD or the catalytic domain and fuse it with the dCas9, then you can actually turn on the BRCA1 gene, and then when it gets turned on, then the cells become more the repair can happen, and then they become more sensitive to the other chemotherapies and all these things. Otherwise, they are not. So some of the other examples of these modulation of expression of the BRCA1 is shown over here, as in this case by using a specific dCas9 fused with a TAT1. So in all this case, the basic principle is same, that you have a gRNA which does the specificity for that where would it be targeted, and then the dCas9, the dead Cas9 is coming, and in which you are fusing the other modulator for the modulation of gene expression. Okay, and then when you do this in this case BRCA1, now one of the which otherwise refractory to these to the chemotherapy, but then when you use the mitomycin C with presence of this gRNA, the cells become sensitive to the growth of the presence of the mitomycin. So chemotherapy, or rather in this case, the drug becomes more effective. Okay. So the PTEN one, I would just skip it because once again it's the same thing. Modulating the expression, modulating the protein, and ultimately modulating the overall proliferation of the cells. Okay. As you can see in this case, when they do the modulation, then whenever you use all these DNA damaging agents or the chemotherapy reagents, then it becomes sensitive, as shown in panel C, D, and B, C, and D. So the overall idea is that modulation of any given specific set of genes, either decreasing expression or increasing expression, so that they become more effective to the treatment of the chemoresistant, because the normal cells, otherwise, the normal cells would be much more sensitive to the chemotherapy than the cancer cells, which is what the main purpose of the chemotherapy regimen is. Okay. Now you can also do a gene editing, in a sense that you can introduce some of these specific mutation if it is there within the gene by the process of homologous recombination. One of those examples are shown over here. In this case, by this transthyretin amyloidosis, which gives the clot, within the clot within the blood vessel. So what they have done is that when they have corrected by the gRNA, then the overall plaque formation gets significantly downregulated. So you can do that editing as well, but the editing is more of a dependent on the homologous recombination that you will have to bring in, okay, along with the gRNA for the editing purpose, but it can be done. Okay. Now, finally, before I end, so this is another example of the disease, the CRISPR-Cas in modulating the other infectious diseases, in this case HIV. Okay, and I'm not going to talk about how the HIV is being, kind of epidemic all throughout the world. Okay, including India. Now, what they have done in this case, that they have actually targeted one of these region of these HIV called the LTR, or the long terminal repeat. So this paper was published about like 6-7 years ago or something. What they have used multiple gRNA to target the LTR. Now, LTR is a promoter, okay, HIV promoter, which helps in overall, with the host machinery, overall transcription of the entire HIV coding sequences. So if you can modulate the LTR, then you can actually think about the overall regulation of the HIV function within the regulation of HIV within the mammalian cells. In this case, the blood cells, CD4 cells. Excuse me. So what happens is that in this case, you target the LTR. Now, one of the big problem of the HIV treatment is that the latency of the overall virus replication. In a sense that the virus gets integrated within the human genome, and thank God the COVID-19 doesn't get integrated, otherwise it would have been a mess for us. Okay, in HIV, the biggest problem is that it gets integrated and then it doesn't come out. And because of which, within the cellular system, sometimes what happens is that because of the treatment of these highly effective, the regimen of the heart and all others, the virus, the pro-virus, what we call, it becomes latent, means it doesn't do anything, just it sits over there. And all of a sudden, when you take out the regimen and all these things, after a few years, few duration, depending on the person to person, then the latent HIV becomes becomes activated, and then once again it gives the pathogenesis. So that's a big problem in terms of curing the HIV infection. So if somehow, if you could actually target those LTR, which is integrated within the chromatin, then perhaps you can think about modulating their expression and then getting rid of those cells which have all this, including the stem cells. Okay, so this is one of those in which they have done, they have targeted gRNA with the LTR, and what they have shown in panel A, the result is shown over here. The reactivation. The reactivation can be done by using one of these called or HDAC inhibitor called the TSA. So when you use the TSA in a normal control cells, you see the green bar type or spike. So those are the reactivation. In a normal cell, there is a huge reactivation, but when you treat it with the LTR specific gRNA, then you see it goes down. So it also, so essentially this CRISPR technology can also be used for modulating the expression of any given gene, or even the LTR, the pathogenesis, which are there within the, which have gotten integrated within the human genome, can be reactivated so that those cells can be taken care by the treatment, by the heart, by the heart treatment and others for the effective care of these pathogenesis. So this is once again talking about the same data, so I'm not going to go into this. So now finally, before I end, so I talked about all this modulation, but it's always easier than done, because the big issue, there are a couple of big issues are there in this technology, and other speakers will talk about it. I'm just giving you a little bit example of this in terms of gene expression and all this. Okay, so the limitations of these technologies. Now, biggest limitation is the delivery system. Okay, what do I mean by that? Is that now majority of the time the delivery is that, first of all, when you are talking about any given specific gene or a cell, cancer cells in vitro, when you are doing within the lab, then you can put the RNA, and then you can put the virus or the delivery reagent within the cells, and you can see the effect of it. But within the human body, you cannot go for all the systematic infections. You have to go for localized one and all this. Now, the problem is that the Cas9. The Cas9 gene is quite big. Okay. Now, majority of the time, this delivery system is a virus mediated. Either adenovirus, which is what normally associated with our cough and cold, and which is what is also being used in all this COVID vaccine and all this with the Covishield. Okay. These adenoviruses act as a delivery system. Okay. Now the adenoviruses, it has a packaging limit, means how much of RNA that it can package. Now, if it has a, let's say, packaging limit of 12 kb, okay, and it has to have its own some of these component, as well as it also has to have your Cas9, which by itself is almost like 9 kb, and then you are fusing that one with your other activator or repressor or whatever that you are thinking about. So these limitations of the packaging is one of the big drawback of this overall delivery of the CRISPR technology within the mammalian cells, or the cells, whatever cells that you are thinking of. Second is that we are always relying on these few bases of the gRNA for the specificity. Now, the overall human genome, if you think about, okay, now having the specific targeting in the specific gene is always challenging, because there is always an effect of off-target effect, which the gRNA goes, and then rather than the one that you are saying, it would also go over there, but also it would go and bind to, let's say, n number of other targets, and then ultimately giving rise to targeting or either activating or repressing those genes, whichever that you are not targeting. So that off-target effect is also an issue. And some of the other speakers might talk about how the overall scientific fraternity are thinking about limiting this off-target effect, so that a specific target-based approach, a specific target-based modulation of gene expression can be done. Now, regarding this delivery, the virus-mediated, There could be one of these examples that I'm giving you, and there are multiple other examples by which it can be delivered. So what people have shown is that by the lipid nanoparticle-based delivery system. In this case, both the effective, the purified Cas9 or Cas9, not the purified Cas9 protein, but that other Cas9 mRNA, as well as the gRNA, can be assembled into a lipid nanoparticle, which can be given to the cancer cells or the tissues. Okay. Now, these formation of this lipid nanoparticle that you can, it's not a length dependent or anything. You can virtually go for any length. Okay. So in that case, the overall limit of the genomic DNA or the Cas9 RNA and others, it becomes irrelevant. So there have some, and one of these examples that I'm giving that was published in PNAS just not that long ago, and they have effectively delivered this lipid nanoparticle based Cas9 and the other gRNA for targeting some of those genes that they have targeted for. In this case, the ANGPTL3 that they have that regulates the expression of the lipoprotein lipase that they have shown as a proof of example. Okay, so with that, I would just end, and then this overall, just before I stop, I must say that this technology is quickly improving, and then we are hoping that in not that long ago, we'll be having a specific modulation of gene expression of specific gene, so that we can regulate the expression as well as the physiology of any given cells for better effective treatment of any given disease that we can think of. And we are hoping that this will be a very bright future, and that's why the Nobel Prize went to them, and it deserves it so. So with that, I would end, and I'd be happy to have any kind of comments or any questions that anyone can have.
Yes, thank you, Devabrata. It was a wonderful lecture session. Especially, you have walked through the basics in the beginning, because I have some very good comments from the participants that you have touched upon the basics, so it was worth it. Obviously, there are many participants from different backgrounds. I do not know whether they have gathered, but your lecture was so basically, the initial phase was so basic that I think they also didn't have any problem. Yes, barring the jargons that you had, but that I think it was fine. So you have touched upon, just to summarize, and then I'll take some questions. What I understand that this is a very important aspect that you touched, that what we have been knowing about the CRISPR-Cas technology, that we simply go in and edit the genome, cutting it, and cutting it maybe somewhere we want, somewhere it doesn't go and cut where we want. We have been understanding that. And also it is important from your lecture that not only the genome editing is being touched, you are trying to use the technology to even increase the gene expression or lowering the gene expression by modulating. And that is something new and something I think many of the participants will appreciate, that this concept was slightly a little bit at the down end. We did not come across such examples. So modulating a gene expression is something that is something really not easy, and it is complicated. And this epigenetic modification is also somewhere is so diverse. But yet you have explained the expression getting controlled. In some cases, you are trying to reduce it, and sometimes you are increasing it in favor of the system. You're trying to build a healthy system by tweaking this arrangement. So am I correct? Did I get it right?
Yeah, absolutely.
Yes. So I think I have some few questions, but before I go into my questions, I will be taking the questions from the audience. Okay.
Sure.
So one by one, I will also ask the participants to raise your hands if you want to directly ask Dr. Bishesh the questions. And some I can see them already on the chat box and in the the question and answer box. So I will go one after the other.
Sure.
So the first question. So the first question that I find from the audience is, okay, at what stage the gene editing is practically possible? At the adult or the 64th stage of embryo? Which one is practically possible?
Well, it first of all, there are, if you ask me which stage would you like to edit the gene, I would go for 64 rather than an adult because it's already fully formed. Okay, so if you want to go for any specific organ or something, you can localized way, you can actually transfer, or localized way, you can put your virus into that, because that medical fraternity has already accepted that you can target a localized organ or something. Okay, but for the whole systems way, if you want to go for, then you have to go for only the 64 or even earlier. But there are ethical issues with it. So let those ethical issues get cleared up, and then we'll think about it.
But it is possible, and it is doable.
Yes, there were examples. There's one of these Chinese scientists.
Oh yes,
yes. Unintentionally, he said, "Yes, I have done it." So.
Yes. Okay. So I have another question. This is from I cannot see the name, but Adesh. Okay, he writes, I hope we have to think of a disease as a multifactorial rather than as a multifactorial to make it more application oriented for its use in human. So
absolutely, Adesh, you are absolutely right. So that's why the this CRISPR screen that we talked about, that would give you a set of genes. Okay. Set of genes, and then set of genes that would be targeted, because no disease is unifactorial or unit. The Mendelian type of disease pathogenesis doesn't really work. Okay, I mean, actually, he was lucky enough to get a single gene-based approach in equally dividing in generations. But that in the disease perspective, it doesn't happen.
Yes,
but you will always have to take a group of genes. So that's why getting the lead or the heat is always important, the effective hit. Okay, and before and then you kind of choose the best out of it to go for a combination of the gRNA, not just a one specific gRNA. And then he is absolutely right. I don't know whether it's a he or she, but the person who has asked for, absolutely right. It's not a unifactorial, it's rather a multifactorial. Yes.
Yes. So what is the fate of the editing after meiosis?
I mean, if you are going for, let's say, the one that I talked about, the activator. Are you talking about the modulation of expression or the editing within the genome?
Editing within the genome after the meiosis. Just
that remains. Editing within the genome, that remains, because you are editing within the genome. So that will faithfully be propagating. Yeah. That will faithfully be propagating. But if you are talking about the gRNA-based modulation of expression, that one, I mean, you will have to keep on treating, because over a period, because those are not integrating. So when the cells will divide, okay, because the daughter cell, one of the cell may have all those gRNA, and because those are not integrating within the chromatin. So one of the daughter cell can have it, but the other cell may not. So you'll have to think about those.
What I understand that it will be, as you rightly pointed out, it would be faithfully, because in meiosis the germ cells are getting edited. So it will be carried over from the one generation to the other, because that is the main carrier. Yes, I think it will be faithfully, definitely. Yes. So ethical issues regarding all this, what is your comment? Of course, we know it, we have heard it, but we would like to hear it from you as well.
I think if you ask me, then I think the scientific imagination doesn't go or doesn't kind of have any barrier to it. So I would restrict it. Otherwise, yeah, there should be some restriction. Otherwise, people would ask only these kind of genes in my childhood, in my children, than on the others, and all this will be a mess.
Right.
Be a mess.
Okay. That's we can understand. Okay. So here, Dr. Chittabrata asks, which technology is more specific and more efficient microRNA mediated gene silencing or CRISPR mediated gene silencing for differentially co-expressed gene sets?
Well, I mean, I would not go into the microRNA one because there is another panelist who would be talking about the microRNA one. So I would leave this comment to him and let him talk about it.
Yes, but also we have to remember that microRNA is not something that we are going to use for editing, because microRNA is an integral part of our system. So it is actually there to regulate gene expression for the system. It is not something to knock out or knock down. It is required. MicroRNA is very much required. So gene expression getting silenced. Yes, please.
Also, the microRNA is mainly for silencing the expression of genes. Okay, not for modulation of expression.
Modulation, exactly. And also it is required because there are certain genes which need to be repressed at a certain time point. Because as we know, and we all know that this microRNAs had been discovered in the C. elegans, and they are there to help in the growth. So some genes would be expressed in the early stages, some at the later stages. So they have to be repressed accordingly. Otherwise, there will not be a proper growth. Yes,
absolutely, absolutely. Yep.
So Sarvajit Kukreja asks, can we use this Cas9 for the treatment of Alzheimer's?
Treatment of Alzheimer's. Yes, I mean, some of the studies have been there, but the Alzheimer's, once again, as I say, first of all, the overall, studies with the neuronal diseases and all these things have been fairly not so, rapidly as we have expected because of the model system and all these things. Those are, so those are not so much available, partly because you are looking at the Alzheimer's when the cells age or the mice age. Okay, so getting the right condition for studying those have been a very big limitations for the Alzheimer's disease studies and all those things. So still do not know the overall mechanism of this, unlike the cancers and all these things, for which faithfully it regulates. So faithfully you can see it within the mouse model and all these things. But for Alzheimer's, those have not been, especially with this, because these are all neuronal impaired testing, and then faithfully doesn't replicate in the mouse system as well. So once we know, then perhaps we can think about it. But until then, targeting the Alzheimer's with the CRISPR would be very premature to comment on it.
Okay, so I have few questions. Maybe they are very basic, but I would like to ask. Maybe it could be a common question for other participants. So, Dr. Bishesh, you have mentioned about this library of guide RNAs, right? Preparing a library of guide RNAs.
Yeah.
How do you do about it? How that library is made? This is the first question. And second is, how you have talked about the different types of cancer. Okay. My question is that as you gradually you are targeting some genes which are expressing at a higher level, and some genes are shown to express at lower level, and you are accordingly targeting your guide RNAs to revert that expression. So my question is, how are you again identifying that whole repertoire of those genes, which is expressing high, which is expressing low, and then you prepare your experiment? Means I am not able to understand how you can start the experiment.
Yeah. So regarding the gRNA, for the gRNA targeting the entire repertoire of the genes, so there are algorithms are there. Okay. Algorithms actually give you this, even for the designing the gRNAs as well. So the algorithms will give you the specific set of gRNA that need to be synthesized, and that takes into account of all the sequences, because the total human genome is known, right? So the machine can give you the best possible gRNA that can be synthesized, and then that can be pulled into the library preparation, like the way for the next generation sequencing is being done, and then you make the virus, and it's a pooled virus, okay, and then you use that virus from the library, from the gRNA to the virus for ultimately ultimately infecting the cells. Now, as far as your targets are concerned, is that, yes, this is always a problem of too many. Okay. Because now, which one to target? Usually, what I have seen is that, although I personally haven't done, but what I have seen, usually they go for the top, let's say 5 or top 10, and then show the kind of proof of concept of these that they are doing. Okay, because if you do not select the top 5, what happens is that then people would say you are not really using the top 5. You are selecting something in between. Then what about the top 5, which give you the best hit? So, so usually they go for, let's say, top 5 or top 10 to make a proof of concept of this overall idea that they are working with. So, but however, at the same time, once again, it becomes like, not necessarily now the one that we see at the end in a paper, that's ultimately a result of maybe 100 different failures. Right? Yes. We all know that. We all know that. So yes. This is the inner story of.
Yes, true. Yeah. So that's what I was wondering. I saw some pictures. It has been taken from papers where the MyoD promoter was used for, I think, the expression. So there were some rat pictures. So the rat the images somewhat thin and mostly they were fat, quite chubby. So is it because that MyoD promoter was used and the muscle expression got, other than the cancer cells? Possibly. Was it? Possibly. Okay. Possibly, yeah. So I was just wondering, because they had picked specific promoters.
Yeah.
So there is one question from the audience. Okay, Shomo Agarwal. Okay. Wait, just a second, please. Can CRISPR technique be utilized for reference in plant biotech, particularly dealing plant diseases? We will have.
Absolutely. There are actually a lot of examples of that. Yes. And most work is going on in the plant system because of its
ethical
issues and all those are not there either.
Yeah. To some extent, that it is safe. And we will definitely, we will have lectures on the plant system.
Absolutely.
So I think Shomo Agarwal can pay attention to those also, so that you get a good idea about it. So any other questions that you have? Maybe let me see whether I have covered my curiosity. Well, okay, one question that you said that. This is a very technical way because we teach students, so I am quite just sort of it came into my mind. You said adenovirus and the packaging is difficult because you are adding so many components other than, because you need a specific size for packaging, and beyond the size it will not get packaged. And below the size it will not get packaged. So there is a limitation on that. Yeah. Now, if you really are keen to use the adeno system, then is it possible to remove some of the elements and make it a little bit smaller so that those genomic DNA of the adeno can be kept and you can add? So is there anything like that they have no problem?
So just to address your things, there are multiple other Cas9 have been found out which are way smaller than the SP-Cas9. In fact, actually, Devajyoti would also talk about some of the shorter version of the Cas9 in his lecture, upcoming lecture.
Yes.
So people are trying to kind of that, the GAL system that earlier people have used, they are trying to shorten it so that the minimal requirement that's there for recognizing the gRNA and the nick function or whatever function. So that depending on, because let's say for the targeting a specific gene, then all what you need, the gRNA recognition, other part you really don't need it. So once people do a very a kind of structure, which is some of those are available with the gRNA with these, then they would come to know that exactly which part would be required for this recognition. Then you chop off the others. Yes. So in that case, the 9 kb would become right now 3 kb. Then you can fuse others. Yeah. So those are going on here.
You're making room for better packaging.
Absolutely. Yeah.
Preeta Mukherjee has a question. What is the post-CRISPR-Cas9 method that should be ideally be used to check for the knockout or knockdown in cancer cells or mouse models, and also in tumorosphere models? So,
okay, this is more of a research question, I would say. So
yes, it
you do RNA analysis. The RNA analysis is not recommended. Usually, if you are going for a deleting a gene, because you are targeting expression of the genes, you do a Western blot to show that those genes are not being expressed. Because RNA you would see expressed, because because it's a in frame, change in the frame. So RNA would be expressed, but would not be translated to give you the protein.
Yes, Western blotting would be the best way to check. Yeah, that is the downstream test.
Downstream, yeah.
Yes, absolutely. So I think we have covered all the questions so far asked, and it was really a very good interactive session. Many participants have they have gained a lot, because it was really a classroom and a research both gelled together, and I really enjoyed it. And thank you so much.
Thank you very much.
And we really appreciate your effort to explain it from a very simple way to going and gradually moving on to the higher level. And on behalf of Macau, West Bengal, I once again thank you very much for your precious time. So our participants will definitely be in contact with you. We will be in contact with you.
Yep.
And whatever sharing of intellectually we can share amongst each other, we will go ahead. And if the participants require any help, I will bother you so that you can help us
no problem.
to help the participants.
Absolutely.
So thank you once again.
It was an honor to present it, and then I really enjoyed, because I like